Test, 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 test. Can you hear me from back here? Yeah. I am actually I know her. One of the former members. Yeah, I'm pretty sure I know her. Was she a postdoc in Oliver lab? Yeah, I covered with. No, I was collaborating with Paul. Just a little bit. And what's her name again? Tara was postdoc in the project. She was supposed to be involved in the project. But eventually, I gave on the project after we moved there. You know, 2005 sometimes teenage and poster with all. Oh yeah. Yeah, I don't know. Because an addition to it, but now in black Harvard, I studied fundamental coders. Oliver was in
Alrighty. Well, welcome everybody. So we are getting kind of kicked off to a new year of Vision Research Seminar Series, and we're excited to have our outside speaker. So Lise is hosting her and can introduce, and then we'll get going. Just a reminder, kind of lunch after for trainees. I think that. I'm already signed up, so you'll get a chance to speak with her as well.
Thank you all for coming. It's really my pleasure to introduce Madineh today. I met Madineh through my journey with transition to a new animal model, the tree shrews, and we spent a lot of time at the Max Planck. She taught me most of the things I know today. She got about tree shrews. She did her PhD at Penn State, then a postdoc at the University of Pennsylvania, and then did her final postdoc at the Max Planck. And now she's also on the track of opening a lab at Cornell, which will officially open its door in January. And today she will take you beyond the retina and talk about complex encoding of location in the visual system. So,
Hi, everyone. Thank you, Elise, for the invitation. I'm really happy to be here
I've had some really fun conversations beyond my comfort zone in the morning. I'm really looking forward to more conversations in the afternoon. Today, I'm going to share with you a story about discovering a strange and complex encoding of spatial location in the visual field and the visual cortex of tree shrews first, and then other animals. If you're not already familiar with these little critters, you will be soon, because Dr. Sedigh here uses them as one of her main model organisms, and she's actually growing an animal colony. They're pretty interesting animals. They have their own order, Scandentia, but they share an ancestral lineage with primates. And like primates, they're highly visual, they're diurnal, and they're extremely agile. But they also share some common features with mice, including their small size, which makes them practical as lab animals. So we think that they're really a good animal to bridge visual studies in rodents and primates and to diversify visual studies in general. The structure of the tree shrew eye is primate-like in some important ways relevant for this particular audience. Like primates, but unlike rodents, the tree shrew optic nerve head has these load-bearing collagen fibers that play a role in the physiology and pathology of the eye, especially in glaucoma. The sclera in the tree shrew eye also contains these collagen microfibers that play a role in the elongation of the eye axis during development. This makes them relevant for myopia and other diseases of the eye. There's the sclera. And probably my favorite feature of their retina is that they're almost entirely made of cones. So even though tree shrews don't have a fovea, they don't have a super dense region in the retina like primates do, their entire retina is full of cones. So here's a plot of 17 different animals showing the ratio of rods over all photoreceptors versus the ratio of rod bipolar cells over all bipolar cells. And tree shrews are this little dot in the left. So they're one of the animals with the lowest fraction of rods or the highest fraction of cones in their retina. And as you probably know, rodents and felines, other model animals of the visual system, have mostly rod-dominant retinas consistent with their behavioral demands and nocturnal lifestyle. Okay, so I know that most of you like to hang out around the eye and the retina, the first stage of the visual system, but today we're gonna jump past that stage. We're also gonna skip the second stage in the visual thalamus and colliculus and go to the back of the head in the visual cortex. And we're going to look at how neurons in the primary and then secondary visual cortex encode or represent different features of visual input, which we control by placing a monitor in front of the animal's eye and then recording brain activity. And I'm going to try not to move too far from this microphone. Okay, so visual cortex neurons exhibit selective responses to various features or properties of visual input. Things like spatial location, so where something is in your visual field, or the orientation of an edge, or the motion of something, or its color. These are all examples of features. In larger brains, in the brains of larger animals, the neurons selective for similar features, like nearby spatial locations, are physically next to each other on the brain. And what this produces is something we call maps. So here's an example of a spatial location map. Here's an example of a map for edge orientation. They're called maps because there's a one-to-one correspondence with the visual input that's shown on the monitor and the activity pattern on the cortex. The most famous map probably is the map for spatial location. It's also referred to as the retinotopy map because the pattern of activation of the retina by visual stimulus coming in from the visual field is preserved on the cortex. So the pattern of activity on the retina matches the pattern of activity on the cortex. The second one is what I already mentioned, the selectivity for edge orientation. Neurons with similar preferences cluster together, forming this pinwheel structure that we call orientation columns. So the working assumption for a long time has been that these two types of feature encodings or maps, spatial location, and then all these other features, edge orientation, color, motion, are independent, meaning that a neuron's preference for a particular spatial location has no bearing on what features it responds to. This is thought to be an efficient coding strategy because it means that the brain has access to all possible features at every point in the visual field, at every spatial location. But if you sort of think about it, this is only an efficient representation strategy if the distribution of features, motion, color, are uniform across the visual field. And we know now, we're starting to know that that's not really the case. And today I'm going to show you plenty of evidence that feature encoding in every stage of the visual system is spatially biased, meaning that a neuron's preference for spatial location is related and can predict its preference for other features. And the particular spatial biases that we see in the visual system are related to the ecologically relevant visual inputs that animals receive in their natural habitat across their lifetime. And I got to this conclusion about the importance of spatial biases and how they can be used to understand the relationship between the brain and the environment of the animal by finding a weird map of spatial location in the visual system of tree shrews and then trying to wrap my head around it, trying to make sense of it. We can measure these maps pretty easily these days. It used to be more difficult using optical imaging coupled with viral calcium indicators. So we inject these viruses, it's a small amount of liquid, into the visual cortex, into the brain of the animals in the right portion, and they enter the cell and produce a change that results in the neurons fluorescing proportional to their firing activity. And then we can record this fluorescence using a video camera that's placed over a cranial window that we've replaced with glass to give us optical access as the animal looks at a monitor that we place in front of its eye. And because the monitor covers most of the visual field of the animal, I'm going to equate what's shown on the monitor with the visual field and with essentially what's projected on the retina. So when I say visual field or monitor location, that's equivalent to location on the retina. And the jargon in our field that I've preserved for historical reasons is to call the horizontal axis of the visual field or retina azimuth and the vertical axis elevation. So that's just a notation point. And we're going to discuss each of these two dimensions separately because as it turns out, the brain encodes them separately. Here you're going to see fluorescence activity recorded in the left and right primary visual cortex of an awake tree shrew looking at a monitor. And it should be clear that the two patterns are related. Let me try that again. There we go. Right, so as the bar moves up, there's a corollary activity pattern in the opposite direction. And if I use a color code now to indicate the regions on the monitor that produce the highest activity in the brain, you can see how the map in V1 is really a simple transform of what's shown on the visual field. If you just take this color map and you flip it and you sort of squish it up a little bit, you get the map on the cortex. And we can use a similar technique to measure the response in the other azimuth direction and the response to other visual features. So that's V1, simple map. What about V2? V2 is the secondary visual cortex. It's basically the next area one synapse away from V1, which is the primary visual cortex that I've been talking about in the back of the head. These are the first and second areas, regions in the cortex that receive and process visual information. So here now I moved the cranial window from overhead a little bit laterally, so it covers V1 and V2 at the same time. And I'll start by showing you the elevation map again so you can anchor yourself to the new window. This is the calcium activity, the white in response to the bar moving up. Here it's at its lowest position, V2 on the left, V1 on the right, and this red bar is the border of these areas. And neurons at this border happen to prefer spatial locations near the vertical meridian, which is the imaginary line separating the left and right visual hemifields. So now as the bar moves up, there's a corollary pattern similar to what you saw across both V1 and V2. And you can see the simple mapping in the colored elevation map, right? In V1, it's beautifully smooth. In V2, it's a little messy, but it's basically similar. So a simple mapping of the vertical dimension of the monitor on V1 and V2. The azimuth responses, horizontal, are very different. As the bar moves from the middle of the screen out to the periphery, the responses in V1 are as expected. There's a single corollary bar, and it basically moves with the bar until it's outside of the window. You can't see it anymore. The responses in V2 are pretty crazy. First of all, there's not a single bar, there's multiple. It's perpendicular to the pattern in V1. It should be vertical, it's horizontal, like the pattern in response to elevation. And the distance between these multiple bars seems to close and open as the bar moves peripherally. So pretty complicated pattern. And notice that this color order and the map now in the color map for the horizontal dimension in V2 reverses direction multiple times. All of these other maps have a single gradient. You go from orange to blue. In V2 for horizontal dimension, you go from blue, that's 20 degrees out, sort of here, back to orange, that's your central visual field directly ahead. And then you go back to blue. So you're sort of going back from center to periphery and back. And it wasn't immediately clear to me what kind of a transform fits between the image that's on the monitor and what the map that's on the cortex. I can no longer just flip it and squish it. But this doesn't have to be a mental exercise because we can actually extract it from the data. So I can understand the transform by drawing a line along sort of the long dimension of V2 here and extracting the positions of the vertical position of the preferred spatial location for these neurons under the line and the preferred horizontal position. And for elevation, the map is smooth. So as you walk across cortex, neurons like similar positions in elevation that sort of just gradually come down, that gradually reduce. For the horizontal dimension, there's this waving back and forth. There's this, we call this a sinusoidal pattern because it looks like a sinusoidal function, where you go from the center of the visual field to the periphery and back to the center. And if we now plot these two things against each other, elevation versus azimuth, to go back into the actual visual field or monitor coordinates, you can really see this complex snaky pattern form. So these central orange and peripheral blue stripes are really a manifestation of this complex snaky back and forth transform. And I'm going to show you an animation that should make it clear how you take the image on the monitor, the visual field, and this transform and get the stripy maps that we saw in V2. So I'm going to start with the map of the visual field on the right, and I'm going to draw the transform. And for each point of the transform, I'm taking a little bar of the same color and putting it in V2. And now you'll see, as I zigzag back and forth, I'm going to get these stripes. And now for the other axis, the vertical axis of the monitor, I'm going to change nothing except just the color map on the monitor is going to be vertical instead of horizontal. And we should get smooth maps, and it should be clear why. Now you're zigzagging along the gradient, so there's no reversals in color. Okay, so this is the sinusoidal transform, and it produces the striped mapping in V2. So the maps in V1 and V2 that I've shown you so far are very different. If we start with these visual field maps again, just to summarize, the map in V1 is essentially, it preserves the retinal image. So if you take a grid and you put it on the monitor and you just transfer each point on the grid, you get the maps in V1. It's basically the same map on the retina along both the horizontal azimuth and the vertical dimensions. If you do the same thing in V2, the transform by nature is now not point by point. You're combining the two axes of the visual field. You're combining the horizontal and vertical And what that produces is basically a one-dimensional long mapping in V2 where the vertical and horizontal dimensions are no longer even perpendicular to each other like they are in V1 and like they are in the visual field. So the sort of interesting question here is, okay, we have this crazy map in V2, we have the simple map in V1. We know that V2 is the next area over. It receives most of its synaptic inputs, most of its visual information from the retina from V1. So the simplest explanation here is that the synaptic connectivity pattern between V1 and V2 is the snaky transform. That's a pretty wild hypothesis, but it turns out to be true. We injected colored tracers in V1 along a line. Under normal scenario of one-to-one connectivity that we expected, you would expect a similar line in V2. But what we got was these stripes. And if you think about the animation I just showed you, this should make sense. What we've basically done is if you imagine this sinusoidal transform, it's going to zigzag into and out of the region of V1 that has viral tracer. And when it passes regions that doesn't, you have these holes. So altogether, it produces the stripy pattern. And this was a really exciting day in the lab because it was basically a sanity check that our imaging results weren't flawed somehow. The anatomy not only supported the functional imaging, but it also explained it. And the fun part was when we looked in the literature, John Kaas's group at Vanderbilt had years ago already reported this strange striped connectivity pattern between V1 and V2, but they didn't have the functional imaging data, so they just couldn't make any sense of it. Yeah.
These stripes correspond to the stripes that Livingstone and Buell discovered.
We will get there. Thank you for that. That's exciting. Yeah, so we really were only able to figure out this anatomy having knowledge of the functional imaging and the sinusoidal transform. So at this point, I was pretty confident in my results, but I figured it's a weird tree shrew thing because I hadn't heard of it before. I hadn't quite made that connection before. And luckily, my postdoc lab also had access to ferrets. So I decided to look at retinotopy maps in V2 of ferrets. I'll skip this. Although the pattern is not as robust as in tree shrews, we found a pretty similar mapping in V2 of ferrets. Here's the map of azimuth. Here's elevation. Elevation is mostly smooth. Azimuth has these stripes, and together they produce a sinusoidal transform in the visual field. And again, when we looked in the literature, we saw that the connectivity pattern had been reported earlier, but without understanding how or why it looked so complex. So now you must be thinking, why? Why does V2 have the sinusoidal transform? Why would it go through the trouble to contort itself so much? Why questions are hard, but we can sort of have some fun and try and come up with some answers. And to come up with some answers, I used a model to simulate retinotopic map development. The model simulates a slab of cortex, a piece of tissue like V2, with a bunch of neurons randomly placed on the cortex. Each neuron, of course, has a preferred location in the visual field, and all of the neurons together map the visual field sort of messily. There are some holes. The goal of the algorithm is to move the preferred spatial locations around so that we cover the visual field very nicely, like this target on the left. The constraints all have to do with the cortex. So the first constraint is that nearby neurons on the cortex have to have nearby preferred locations in the visual field. The second is just like V2, the border has to represent the neurons at the border of this area, have to represent the vertical meridian. And the third is that the shape of the preferred spatial location, the receptive field, for those of you who know what that means, can be too stretched. And these are all biologically imposed constraints. And that's premature. Now, if I run the simulation, you'll see that, you will see that the visual fields first collapse, and then they fill back out and produce nice smooth maps. And then I'm going to change nothing about the model, except that I'm going to elongate this piece of tissue, the total area and the number of neurons, and everything else will stay the same. And now you'll see that the map folds in on itself as it tries to cover the visual field because it's constrained by the boundaries of this elongated region. The elevation map remains smooth because it's anchored by the vertical meridian. So V2 has this sinusoidal transform because it's an optimal representation of spatial location when the cortical area is elongated. But that's not a really satisfactory answer because it just passes the buck. Why is V2 elongated in the first place? And it's not just elongated in tree shrews, it's elongated in many animals, primates and cats and ferrets. So there's got to be a better explanation. I have some ideas that we can discuss if there's interest later in the Q&A. Okay, so far we've only talked about retinotopy maps of spatial location. But remember, our goal was to understand the relationship between the retinotopy maps and the mapping of other features like orientation. So in a second set of animals, we mapped both retinotopy and a few other features like edge orientation. And we found that to various degrees, these maps were related. So knowledge of the retinotopy map or the preferred spatial location of a neuron could predict the spatial layout of these other feature maps because they co-varied on the surface of V2. The maps were correlated. Here you can see the retinotopy map in V2, and that neurons in these yellow-orange regions that prefer the central visual field exhibit clustered orientation columns much more robustly than neurons in the blue stripes that represent more peripheral locations. We also looked at two other features, binocularity, whether a neuron responds to inputs, visual inputs from one eye or both eyes, and retinal disparity, whether a neuron is responsive to a cue for 3D depth that comes from the disparity in images because your eyes are horizontally offset in your head, or whether the neuron likes flat images. And all of these features had a clear correlation with the retinotopy map. Here I'm drawing these traces in each color, represent the magnitude of the response to these different features across the cortex. And this sinusoid is the preferred horizontal or azimuth location. And you can see that the shapes are the same. They're anti-correlated just because the central locations of azimuth happen to be represented with lower numbers, whereas higher response magnitudes are represented with higher numbers. So these two things are very much correlated. So to summarize this, we found striped retinotopy maps produced by a sinusoidal transform in the tree shrew and ferret V2. We also saw that the feature maps were also striped and correlated with the retinotopy map. And here's where cross-species comparisons come into play. Striped feature maps in V2 were already famous and enigmatic because they had been discovered to much acclaim in the 1980s in primates. So if you look at the maps for, say, orientation in primate V2, it looks like this. So we thought, could the same underlying retinotopy pattern, namely striped, explain the striped feature maps observed in private V2? And you might be asking yourself, well, didn't the monkey researchers already look at retinotopy maps? In general, recording any responses in monkey V2 is a difficult and heroic task because monkey brains, just like human brains, are folded, and much of V2 is actually stuck in a fold. So you have to sort of stick electrodes in at very difficult angles, and your spatial resolution is low. But looking back at the old data, there's some hints of a sinusoidal transform. So here we can do this again in the tree shrew. If I move across the stripes in the azimuth or horizontal map of visual space, we get the sinusoid going back between center and periphery. And the same data in primates is shown here, cortical distance as a function of the position, the horizontal position of the spatial location. And you can see it's sort of oscillating back and forth. It could possibly be a sinusoid, but it was interpreted to be continuous and linear progressions of spatial location that suddenly reset at the border between stripes. And I think an alternative, and this was important because this resetting of the retinotopy map was sought to ensure that spatial location and feature preferences were independent. So there were 3 copies of the visual field, 1 for orientation, 1 for color, 1 for the other feature that was represented. In light of our findings, I think the sinusoidal transform is an alternate explanation, which means that location and feature preferences are very much correlated. And once calcium imaging works in primates, it's still difficult. We can actually test this hypothesis. So why does it matter if striped feature maps in primate V2 are correlated with the striped retinotopy map? It's important because of the critical role that V2 plays in relaying information from V1 to the rest of the brain. The visual system, some of you might know, is divided into 2 parallel streams, the ventral stream and the dorsal stream. And these streams are related to different behavior. So the ventral stream is involved in object recognition, face recognition, generally recognizing features and things that don't move, static object recognition. The dorsal stream is related to actions and generally involved with faster motions. It's known as the where pathway. The ventral stream is known as the what pathway. V2 is the first separation point of these 2 pathways. And different stripes in V2 feed either the ventral or dorsal stream. So in saying that V2 feature stripes are correlated with V2 retinotopic stripes, I'm saying that the inputs to the dorsal and ventral stream have different spatial biases. Maybe the central stripes that represent the central visual field go to the ventral stream that does things like face recognition, and the more peripheral stripes feed the dorsal stream that has to do with our actions and self motions. And, you know, why would V2 be gating different spatial information from V1 to the rest of the visual system? That would only make sense if these 2 parallel streams themselves had spatial biases related to the functions they carry out. And this seems to be the case. And there's, if you look in the literature, there's mounting evidence. For instance, here's an example fMRI data set from a human. This is a digitally flattened human brain. In a patient that's responding to visual images. On the left, you can see pixels that are responding to either faces in orange or buildings in blue. On the right, it's that same person responding to either the central, the very foveal portions of the visual field in yellow, or the peripheral portions in green. And you can see that the face regions, face-sensitive regions, tend to be near the foveal representations, and the buildings are more sort of in the periphery. And if you think about this, it makes sense. So the point is, at different points in the visual system, early on in V2, later on in these higher areas, feature sensitivity comes with spatial location biases that make sense if you think about the behavior of the animal. Yeah.
Has that been sort of formalized that, for example, recognition of buildings been presented parafoveally versus foveally?
I would say no. So we wrote this review paper in an attempt to really formalize this because there's all these lines of evidence. But if you look in any textbook, the textbook is still parallel. These parallel streams have the same spatial biases. Yeah, thank you. How am I doing on time? Okay, so we can summarize these not quite formalized recent evidence like this. The ventral stream, which deals with face recognition or sort of far space, has an upper and central visual field bias. The dorsal stream that has to do with our actions and self motions has a lower and peripheral bias. And altogether, the lesson here is that there are prominent spatial biases in the structure and function of the visual system, and this likely reflects prominent spatial biases in the visual experience of the animals over their lifetime. For example, as primates, we are social creatures and face recognition is really important to us. So the ventral stream that handles face recognition isn't going to waste resources representing peripheral locations where faces don't often appear. I guess unless you're on Zoom, it's gonna spend its resources representing the central visual field where faces that we are attending to and speaking with most often are. And for other species that are less social, the spatial biases should be different. And in fact, they are. Mice aren't that social, and they don't have a phobia, and they don't have face recognition regions. Their entire retina is sort of like our peripheral visual field, it's low resolution. But the visual inputs they receive are very different between the upper and lower parts of the visual field because they're low-lying prey animals, and there's a lot of threats and sky overhead and terrain and food below. And if you look at the spatial biases in their parallel streams, which were recently defined, it's split across lower and upper visual field biases and not central and peripheral. So this brings me to the research program in my new lab. I think what I learned from all this is that the statistics and biases in visual inputs that an animal experiences that are relevant to the animal are important in understanding the structure and function of the visual system. And these biases are shaped by two things: by the environment of the animal, and importantly, by the animal's own movements. And while we've looked at the environment factor for many decades, we've asked questions like, how does the natural image statistics of this animal relate to its feature encoding? We haven't really looked at how movements shape visual inputs and therefore shape the visual system of the animal. And this is, I think, a really missed opportunity, because while we can't really access the natural environment of most of our lab animals, we can access fairly easily a proxy of their natural movements in the lab. So as an example, there's the subfield of visual neuroscience that I find very interesting that tries to make sense of the very different, diverse layout of retinal ganglion cells in the retina. So they're not uniformly distributed. In primates and humans, there's a very dense region in the fovea, and it falls off from there. In other animals, the shape of this density is very different. And people have been trying to make sense of these differences as a function of the natural environment of the animal. Is it a low-lying animal? Does it need to keep the horizon? Are there trees nearby? And although this is super interesting, these are hard hypotheses to test. To test them, we need to record the natural visual experience of animals in their native habitat using basically GoPro cameras on their heads. But in addition to making sense of retinal structure or any other visual structure by looking at the environment, we also need to look at their movement patterns and the changes that species-specific movement patterns produce in retinal inputs. So for instance, we can ask the same question and try to maybe ask, okay, well, as the animal moves, let's say in a particular behavior, is there a region in the retina that's most stable, that undergoes the least amount of retinal slip? Maybe you want to put the really dense retinal ganglion cells right there, because that's sort of the highest resolution, most stable part of the retina. The point is we can use a cross-species approach with carefully selected animals to understand how species-specific movements shape the structure and function of the visual system. And to really drive this point home, here's a slowed down video of a tree shrew doing a very routine jump and flip behavior. They do this probably 100 times a day, so long as they have enough room in the cage. And if you just kind of imagine the visual inputs experienced during these very routine jumps, a rat that crawls or even a cat is never going to experience these visual inputs. And we know it stands to reason that the visual system of tree shrews has adapted to this visual input, but we really don't know how. So to understand how movements influence visual inputs and later visual processing, we need to measure both the movements and the visual inputs that they create using GoPro cameras. So that's what we've been setting up, a first draft of this. So on the left, you're looking at the animal in its home cage. On the lower left, very obviously, is this chaotic input stream because the animals move so fast. And to make sense of this data set, we need to do a couple of things. We need to A, quantify their behavior. And we do this with existing softwares that use machine learning techniques to track the different points on the animal's body. So for instance, here's a jump. This animal did 4 more of these jumps in a very short time frame. And this plot is showing the position of its different body parts in these 4 jumps. And you can see that it's very stereotyped. These traces are all very close to each other. And then once we have the movements quantified, we can use a computational model to couple the movements with the visual inputs from the head-mounted camera to really understand how are movements influencing the visual input, and then set up some hypotheses for how neural networks should adapt to these inputs. And lastly, we're setting up all of this sort of wireless, free behaving paradigms, but will always be limited in the data that we can collect. So we're also using eye movements as sort of the only body movement that we can study in head-fixed animals, which allow us to keep doing calcium imaging and record neural activity in hundreds of neurons at the same time. So here the question is, how do neurons in the visual cortex represent movements generated by the animal via eye movements versus movements that we impose on the animal on the monitor by just moving a dot or a bar. So that's it. I'm hiring. So if you know anyone that is interested in any of these questions, please send them my way. And thank you for your time and attention. Thanks. And I just want to point out, David Fitzpatrick was my postdoc PI who I did this work with at Max Planck, Florida. He's a big proponent of tree shrews. He's the reason Elise and I have been able to get our labs off the ground. And Susan Frelling is the tree shrew caretaker at Max Planck, and none of this work would have been possible without her. And I'm happy to take questions. I'd love to start with questions from students.
Hi.
Perfect.
That was a really great talk. I have 2 questions. My first one being, like, when did like the V2 as a processing area start to show up in animals? Are there animals that have it and really where like that?
Yeah, that's a great question. When did V2 in the evolutionary time scale show up as a cortical processing area, and are there animals that don't have it? Yeah, so mice don't have a proper V2. They have this ring of secondary visual areas, but they don't have the long region. And there's some theories as to why. One might be just different convergent evolutions, but the other theory is that because their brains are small, you have different processing needs, and so maybe you need less visual areas. But other than rodents, all of the other mammals that we've studied do have a proper V2, so it evolved pretty early, and it may be from a common ancestor, or it may have converged multiple times.
And my second question was, for the tree shrews, since they'll be limited by the cage space, how do you think putting that movement is going to be to visual bias?
Yeah, that's a really good question. That's a tough question because they live in trees and they like to jump from one tree branch to another tree branch. So we use large cages. They are expensive, and that it restricts their movements still. So my plan is to actually use that constraint by designing a cage whose shape can be changed on the fly. It can be flat to make the animals crawl to really prevent their jumps, or it can be volumetric and much bigger than what we have to allow movements. And if we do this very early on in development and rear animals this way, that might give us some insight into, okay, well, the animals that are reared this way can't make proper jumps, or they have less neurons that are sensitive to really fast motions. But it's a tough problem. Yeah.
This idea that the tree shrew will jump and then be upside down, presumably most of its life is right side up. You talked about correlating the environment with biases and which parts of the visual field
Yes.
will be linked to processing horizon versus ground for a a rodent where they're looking for predators, above, et cetera. So this strikes me as really interesting because here you've got an animal that most of the time has a difference in, up versus down, what it's trying to perceive, but then literally flips, 180 degrees
Yeah.
and needs to navigate. So I don't have a question on it, but it just seems.
I think I get what you're saying, like, would everything just average out if they're flipping? Yeah, I've actually thought about that a lot. And I think there's sort of two possibilities. And to tease them apart, what we have to do is to really quantify the sub movements of the animal and separate, say, flipping behavior from when the animal's just sitting there with a right side up visual field. Because what's likely is, okay, maybe on average, there's some overall pattern that persists, but what's likely is that the visual demands are actually changing on the fly. So when the animal is making these very fast flips, it might be, for instance, that it's not really doing much visual processing. Maybe it's not really seeing the fast motion, it blocks it out, or maybe it's not seeing color because all it needs to know is enough to land. So I think the solution to that very challenging problem is to really understand sub-syllables of behavior and approach the data analysis that way.
Is V2 strictly downstream only from V1? Is parallel processing that inputs from the retina into V2?
Yeah, nothing is ever strict in the brain. There are not direct retinal inputs as far as we know. There have been reports of LGN inputs to V2, but there's all sorts, there are, but there's pretty dense feedback pathways. So from higher visual areas beyond V2 back into V2, and some of those areas receive collicular inputs. So there might be a feedback pathway from the retina to the colliculus back to V2.
And I may follow up.
Yeah.
Can you elaborate a little over visual midbrain in these animals because it's substantial. It's not primate bone.
You're talking about their visual thalamus, their LGN,
No, I'm talking about
the colliculus, yeah. Yes, yeah. So I think we have to look at the colliculus sooner or later, because if you're talking about any sort of subconscious visual motor orientation, landing, that sort of behavior is mostly in the superior colliculus of these animals and most other animals, especially small animals that move a lot. I'm starting in the cortex, largely because I've done all of my studies in the cortex and because it's extremely accessible. The brains of tree shrews are smooth, so if you just put a cranial window, you have access to multiple cortical visual areas. The superior colliculus is deeper. You have to, this is Elisa's expertise, you have to use a prism to wedge the cortex away so you can access the superior colliculus. Some of the freely moving EPHIS plans that we have involve the superior colliculus, hopefully in collaboration with Elise. So yes, we have to go there. It's very important. I'm just not there yet.
Do you think it's valid to think of V2 as having multiple copies of the horizontal visual field?
Yeah. Thank you for asking that. I
Some
love
sort of
this question.
divergence, such
Yeah.
as, one analogy in the retina would be one cone signaling to 12 types of bipolar cells, right?
Mm-hmm.
So representing that one local point in the visual field by using 12 different types of bipolar cells. This may be kind of similar to that. V1 signaling to multiple copies of the horizontal visual field within V2.
Yes, I think multiple copies of the horizontal portion of the visual field. I think that's almost certainly true. We see it in these reversals. You go from the center to periphery. But I would say we know there aren't multiple copies, multiple representations of the visual field in general, because when you look at elevation and azimuth together, you really only have one copy. What you're doing is snaking across the whole visual field, but you could interpret it as, for instance, the central and peripheral regions are very important to communicate with different biases to different parts of the brain. So you need multiple copies to make the wiring short between V1 and the rest of the visual system.
So you think that maybe the multiple copies, they do different things?
No, I think they do the same things. I think they send the same information to different regions of the visual system. And it's sort of an efficient short wire. The reason V2 is elongated and has the sinusoidal transform is to enable the brain to send different spatial information from V1 to the rest of the visual system without having to go all the way back to peripheral V1, which is in the back of the head.
So the analogy, my analogy was wrong then. It's not 12 types of bipolar cells analyzing the same point in 12 different ways.
Right,
That's incorrect.
yes.
How many copies do you think there are? You're
We-
showing, it looks like only 2 here, but
Yeah, we've looked in the
more
tree
like
crews.
6 or 7, right?
Yeah, you can tell in the anatomy because this is almost all of B1, so it's depending on the animal between 5 or 6 units. Did that answer your question?
Even though there are only two streams, right? Dorsal stream.
Yeah,
ventral
even though there's
stream.
only 2 streams. So in primates also, there's many more stripes in primates actually. I think some of that might have to do with the size of V1 and the size of the rest of the brain. So if V1 is large, you need a thin area, and the thinner the area, the more stripes you have. That's what our model told us. Maybe that's why you have so many stripes. This is conjecture, of course.
Yeah, this is kind of a two-parter. My first component is I wanted to ask if the weighted spatial biases seen in cortex due to the azimuth and I guess or elevation axes change as a result of head motion or rotation itself. And then if you know if there's some kind of mechanism to I guess adjust these biases as a result of correctivity.
Yeah, that's actually one of the other sort of underlying hypotheses I had. If you think about the simple map in V1, and if you ask yourself what pattern on the monitor or visual field would sort of produce a continuous activation pattern on the cortex, which would maybe make some things more efficient or simpler. It's just a line or a bar that's moving in this way or that way, a very simple linear motion. If you ask that same question for V2, the pattern in the visual inputs that would activate V2 in a continuous fashion looks like this. It's that sinusoidal transform. And if I think about the visual experience of tree shrews, which admittedly I don't know very well, I haven't been in the Malaysian forest, I can't think of anything that moves like this. But tree shrews, if you look at them in Elisa's colony, they do have these they basically saccade with their heads. And the saccades are very prominent in the horizontal dimension, not so much in the vertical. So it could be, it's another very hard question to test because it's not like you can raise an animal that doesn't move its head. It could be that this representation is also related to these head movements of the animals. Because these head movements and the representation, what they do is bring together different parts of the visual field that would normally be far on the cortex nearby. So this is useful for background segmentation or other things like that. Did that answer your question?
Sorry.
Sorry, go ahead.
So going along like with the movement relationship, when we're looking at the tree shoes and then jumping and inverting in that relationship, I'm wondering if there's any dependency or like bias on that consistency of motion. So if they're jumping the same way, is that relationship based on like, are you seeing that kind of more because they're repeating that same motion? Or if we had a tree shrew like jumping and inverting in a different way, like almost somersaulting, would that relationship stay consistent in terms of like how they're moving their body?
Yeah. I don't know. I think to answer that, you can do 2 things. You can either really quantify, like I was saying, the sub movements of behavior and make some predictions, and then in the animal that's actually jumping that way, record neural activity and test it. If you put water bottles in the cage to force the animal to change its path, or if you have something that comes out every other path, does the animal change its behavior, and does the visual responses in the visual cortex sort of adapt to that on the fly? Or you can change the relationship between the movements of the animal and the visual feedback, the visual inputs, using prism goggles that say flip the visual field upside down or left to right. And that'll also sort of allow you some entry into that question, which is, if you change these relationships, does the brain adapt on the fly, or does it not? Is it sort of hardwired for the stereotype behavior? And when the animal goes off that, the brain can't adapt. My guess would be that there is on the fly adaptation, because think about it, if you hurt your leg and you're on crutches for a while, it's not like you suddenly, don't understand the world. Your brain has to adapt pretty quickly to changes in your body. But that's a long-term goal. Great question.
It's beautiful work.
Thank you.
Beautifully presented also. My question is, might be naive, but you showed the bar moving when you're doing your mapping. You showed it moving very slow. You ever vary that speed? And if you do you get the same thing?
Yes. If you vary the speed to a certain point, you get the same transform. If you go past that, if it's either moving too slowly isn't a problem. In fact, we actually did flash bars and flash squares to make sure that the mapping that we saw wasn't somehow a function of the visual stimulus. If you move it too fast, you can go beyond the threshold where the animal's actually seeing it. And also the calcium responses are sort of slow. They're slower than actual activity. So either the animal might not see it, or the calcium activity might be too slow to indicate it.
But within those thresholds, it's independent.
Yes, it's independent.
You need to be mindful of the monitor's refresh rate, right?
Yeah, that's always really important. Yeah. And we're much slower than all of that. That's a good question. For other studies, it comes into play.
What's
So my question, I'm a disease-oriented person, so so my question is kind of in disease states, what is the plasticity of the retinotopic maps? And will they
Overall,
rewire?
large scale plasticity, it's actually pretty small. So if you look at congenitally blind people who have never experienced photons in their visual system, their retinotopic maps are intact. And that the thought is that the maps are actually formed from multisensory inputs because they're sort of related to higher tasks, not particularly just visual inputs. Yeah, so retinotopic plasticity.
So in terms of some, like, let's
Yeah.
say you had a tree shrew that was missing superior visual fields for
Yeah.
both eyes, like you just get the lids a little bit lower.
Yeah, I think eventually you could get some filling. Yeah, yeah. Yeah. But they don't change beyond filling, right?
The shape doesn't change.
The shape doesn't
So
change.
just compression expansion.
The big picture question. You guys are working on tree shrews. Tell me, and I understand it's the closest thing sort of finding all these other what's the advantage of it over squirrels?
Yeah.
And choice for
The squirrel visual system isn't as similar to the primate visual system, especially its visual cortex and LGN, as tree shrews are. So squirrels don't have orientation columns. And, for sort of random historical reasons, orientation columns are a very important thing to visual scientists. Their LGN isn't 6-layered like the LGN of primates and tree shrews. Elise is telling me it's 3-layered. Yeah. 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 Yeah, I don't want to think about that. That's a whole other challenge. The advantage, I will say the advantage over small primates. So marmosets also have a smooth brain. They're also small. You can also keep them in similar sized cages. They have a long gestation. So the gestation of these guys is 40 days. For marmosets, it's 150 days. So you can build a colony quickly. All right, thank you guys.